Take over, Viviane. Thank you. Hope everyone enjoys.
Thank you so much. All right. So good afternoon, everyone, and thank you all for joining today to this online webinar, and thanks for the team in Liverpool for the invitation. I'm very excited to be here. So I've been now at the University of Tel Aviv for about a year. I recently established my own ancient DNA group, an ancient DNA lab, where we'll be working on studying ancient populations using genetic material. And before that, I was a PhD student and then a postdoc at the Max Planck Institute for Evolutionary Anthropology in Leipzig in Germany, where I conducted much of the research that I'll be talking about, or at least the parts that I've been involved in. And what I'd like to talk to you today about is how we can use sediments deposited at archaeological excavations as a source material to retrieve ancient DNA, ancient genetic information, to learn about our evolutionary past. So I'm sure this comes as no surprise to many of you working in the fields of ancient human research, that ancient human genomics has been undergoing a huge expansion in the last few years. If only in 2009, we had no ancient human genome that had been sequenced, we have now today over 6,000 individuals, ancient individuals whose genome have been recovered and reconstructed, at least to some extent. And while the majority of these come from places and dates that are younger than 10,000 years, we are getting more and more of these genetic data, these genome-wide data from ancient individuals dated to the late Pleistocene as well. However, in many ways, our ability to recover ancient genomes and to conduct ancient human genomics analyses is dependent on the archaeological record. And what do I mean by that? I mean simply that there is a characteristics of prehistoric excavations that rule the fact of whether we can or we cannot find human remains and use them for genetic analyses. So just to make this point, I'd like to take you on a little virtual tour at a typical excavation from a prehistoric site. And as I'm sure many of you know, if you would be excavating in a late Pleistocene or middle Pleistocene context, what you most often find are lithics materials, stone tools, that the people that were living in the site or doing activities at the site left behind. You're also likely to find a lot of animal bones in many cases. Some of them will be bearing traces of having been butchered or otherwise manipulated by the ancient humans. And then only maybe, just maybe, you'll find one, two, or a handful of remains of the hominids themselves. And I'm showing you here a beautiful skull, but of course, in many cases, we'll find fragments that are much smaller and even sometimes not indicative for the human remains themselves. And even if human remains are found, they often represent a single or a few discrete points in time and usually do not cover the full stratigraphy, the full time span that an archaeological site can cover. But of course, if you think about it, the thing that you find the most at these types of excavations is simply the sediment, where all these other artifacts, all these other findings are found. And interestingly, the fact that DNA can survive in sediments, both plant DNA and animal DNA can survive in sediments even over hundreds of thousands of years, has actually been known since 2003. And this is a figure from the pioneering paper that talked about recovering ancient DNA in sediments, where the group led by Viviane Slon looked into permafrost cores from Siberia, ranging to about 10,000 years ago to over 400,000 years ago, and looked for DNA in them. And they were able to recover a multitude of different plant taxa, as well as extinct megafaunal species. And they were also in the same paper, they showed that they can also do this in sediments from more temperate environments. So they collected sediments from New Zealand and were able to find DNA of extinct taxa, extinct birds, and also a wide variety of plants. And this ability to recover DNA from sediments has then been applied, I would say, quite widely on natural environments and sort of natural contexts. So, for example, lakes or glaciers and so on. This is another example of a work that was conducted this time in Alaska, also on permafrost samples, where by retrieving DNA from the sediment samples, the team was able to show the late survival of both mammoths and horses in Alaska for thousands of years later than was previously thought. So that's one example. And another example that just came out, I think 2 weeks ago in Nature, a very large study encompassing 500 samples of both lakes and permafrost in the Arctic, in several sites in the Arctic, spanning a time frame of 50,000 years, where they were able, this is a study again from the Viviane Slon lab, where they were able to look into variations into plants and animal diversity over this very large timescale in the Arctic, and also trying to understand the extent of the human impact on the observed biodiversity. However, there was little application of these sedimentary DNA analyses to archaeological context. Among others, because of the difficulty, especially if we're thinking of working with ancient hominin or ancient human DNA, the difficulty of telling apart ancient DNA from modern contaminating DNA that is introduced into the samples. And so back in the days of my PhD work, we wanted to see if by using what was then state-of-the-art methodologies of ancient DNA and phylogenetics in general, we can try and find a way to adapt these methods that we were applying to skeletal remains at archaeological sites. If we can find a way to do this also for sediments, if we can find a way to retrieve, identify, and authenticate ancient hominin DNA from these sediments, even and especially even in cases where skeletal remains are not found. So in this pilot study, we collected 85 sediment samples from 7 archaeological sites in Eurasia, and all of these are sites where it was known from either the archaeological record or the anthropological record that ancient hominins were once present at these sites. And the sediment samples spanned between middle Pleistocene layers and late Pleistocene layers. Then in the lab, what we do is that we take a small subsample of the sediment and we extract the DNA out of it and prepare what is called a DNA library. And at this point, we have in our DNA library a mixture of all the DNA that was found in the sediment. And that is a mixture of mostly microbial DNA of microbes that live in the soil, but also plant DNA and animal DNA, and maybe hopefully in all this mix, also human DNA. And so going directly into sequencing this sort of DNA library can be very expensive because you would be sequencing mostly microbes for most of the samples that you would be looking at. And so with this idea of trying to look more specifically into the human component in these libraries, what we did is another laboratory step called capture, in which we use probes that span the mitochondrial genome of 240 different mammalian species. And this is sort of just to give you a sketch of what that looks like. So each probe represents a part of the mitochondrial genome of one mammalian species, including humans. And we can use then these probes to sort of physically fish out from our library only the DNA fragments that bear a resemblance to a mammalian mitochondrial genome, and then sequence only those. We then take these fragments and we compare them to a larger database of faunal DNA of known genomes to try and tell apart for each of these DNA fragments which animal species does it come from. Now at this point, we could still have a mixture of both ancient DNA and modern contaminating DNA in our samples. And so to tell these apart and be able to do this very crucial authentication step between modern and ancient DNA, we have to rely on characteristics that differentiate modern from ancient DNA. So first of all, we know that DNA over time breaks down, undergoes fragmentation, so that ancient DNA fragments tend to be much shorter than modern DNA. But perhaps more indicatively, we also know that there are specific damage patterns that accumulate over time in ancient DNA. Specifically, cytosine bases, especially when these are at the ends of DNA fragments, tend to undergo chemical change and become another base, uracil, which is then read by PCRs when you're amplifying your library as thymine. So that causes a typical C to T changes at the ends of genuinely ancient DNA fragment that you don't see in modern DNA. So just to show you what this would look like, this is an example of one sediment sample from one of the cave sites that we worked on in this pilot study. And when we looked at the hyena DNA that we extracted from that sample, we saw that there was this elevation of the C to T changes that I just mentioned at the beginnings of fragments and at the ends of fragments. And this is exactly what you would expect to see if you were to take a hyena bone from 50,000 years ago and sequence it. But in contrast, in the same sample, we then looked at the human fragments and we saw, well, absolutely nothing, essentially a flat line. And that tells us that for this specific sample, while the hyena sequences were ancient, the human DNA was not and was stemming from contamination. And what we did was to do this test of looking for these damage patterns for every single taxon in every single sample that we processed to be able to tell where we have genuinely ancient DNA. And so when we did that, we realized that we were indeed able to recover ancient DNA in archaeological sediments in 6 out of the 7 sites that we've investigated. The one site where this did not work is Condé-Larago, which is a site in southern France that is older by several hundreds of thousands of years compared to the other sites that were included in the study. So perhaps where DNA preservation was anyway expected to be very, very poor. But overall, in late Pleistocene layers, we had quite a high success rate. And even we also found ancient DNA in the Denisova cave in Siberia, also in middle Pleistocene layers. Now, specifically as to what type of taxa we were able to identify, were we able to see ancient faunal DNA from 12 different taxonomic families, and that including taxa that are now extinct. So we had some woolly mammoth and some bears, some cave bears, some cave hyenas, and so on. So this sort of gives a good further authentication that the DNA that we recovered was indeed ancient. And just to give you an idea, sort of, for example, wherever we found elephant DNA, we saw that it best matched the woolly mammoth mitochondrial DNA. We did not find, as would not be expected, any modern elephants. But we could also see, for example, that for the bears, we were able to identify different types of bears from the DNA that we recovered in these archaeological sediments. So in the Denisova cave in Siberia, we found mostly brown bear mitochondrial DNA. Whereas in Vindija cave in present-day Croatia, we found the eastern type of cave bears. And this is what types of bear that one expects from the zooarchaeological and previous genetic work that had been done at these sites. But I did mention that what we were looking for was ancient hominin mitochondrial DNA. And at this stage of the study, we had only identified ancient hominin mitochondrial DNA at one site, El Cidrón in Spain. Whereas you'll see from the figure, we also did not find DNA of any other animal. And that led us to think that perhaps the reason why we're seeing hominin mitochondrial DNA in El Cidrón and not everywhere else is because El Cidrón is quite unusual in the archaeological record. It's made out of a single layer that has dozens and dozens of Neanderthal remains, but very few animal bones. And so we thought perhaps, we're seeing DNA of hominins in El Cidrón, but not in other places, because at all these other sites, there's so much of an abundance of animal DNA that it could be overshadowing whatever smaller traces of DNA the humans could have left behind. So with this in mind, we went back into the lab, and this time we performed capture using these probes that are specific for human mitochondrial DNA. And then we sequence only those fragments. And when we did that, we saw that we were able to identify ancient hominin DNA, not only in El Sidron, but also in 3 additional sites. And that got us very excited, and we went back to these samples and generated as much data as we could from these samples. And we were then able to reconstruct for 9 of them between partial and full mitochondrial DNA genome sequences just from sampling sediments. And we could then compare these to mitochondrial genome sequences that had previously been reconstructed from skeletal remains. And when we did that, we saw that for 8 of the sediment samples, the DNA in the sediments fell within the variation of Neanderthal mitochondrial DNA, shown here in red. But for 1 sample taken from the Denisova cave, we found sedimentary DNA that matched the Denisovan mitochondrial DNA, showing that by sampling 9 sediment samples from 4 archaeological sites, we could retrace a large part of the known genetic diversity of human mitochondria in the Pleistocene that had previously been known from skeletal remains. And so we found Neanderthal mitochondrial DNA in these layers that are very rich in skeletal remains, the Sidron that I already mentioned, but this is also the case in Chakilskaya cave in Siberia, where remains of several Neanderthals had already been found there. But we could also do this in layers where there are no skeletal remains. So this is a sketch of the stratigraphy of one of the chambers in Denisova cave. And what we saw is that we found Neanderthal mitochondrial DNA in the sediments, shown here by this little shovel, in the same layer where previously a Neanderthal bone had been found. But we also found Neanderthal as well as Denisovan mitochondrial DNA in layers without any skeletal remains, lower in the stratigraphy than any of the skeletal remains that had been found, showing the presence of these ancient hominin groups at the site much earlier than was previously thought. And at this point, I have to say that this specific sample that had the Neanderthal DNA sort of quite early, quite deep in the stratigraphy, was later reevaluated as part of a new study into sediment DNA in Denisova cave, and this is something I'll talk about later in the talk. And it was found that this sediment sample was actually redeposited from higher up in the stratigraphy. So the Neanderthal part didn't hold up, that's science. But I would say that the Denisovans, sort of the early arrival of Denisovans, does hold up in repeated studies. And so this is Denisova cave, where we had already known that there were skeletal remains of both Neanderthals and Denisovans, but also at Toelves, a site in Belgium, we found Neanderthal mitochondrial DNA in layers where there were no hominin fossils at all, and where it had been inferred that Neanderthals were present based on the types of stone tools that had been found there. So overall, this told us, this pilot study, that it is possible using this methodology to recover ancient hominin mitochondrial DNA from archaeological sediments, and we've shown that for Neanderthals and for Denisovans. Now, what about ancient human DNA? So to talk about that, I'm going to take you to another site. We're still in France. This is at Plateau, and this is a site that has been excavated already from the 70s, where there's a very long stratigraphy, as you can see here from the figure, spanning multiple archaeological layers that go from the early Aurignacian through the Gravettian and up to the Solitrean. And where, incidentally, and that has nothing to do with my work, but it's, I think, a nice figure, this Venus was found and dated to about 21,000 years ago. So an exciting site to work and to look at possibly differences in human DNA and human populations over time. And what we were able to work on are some sediment samples that had been collected in the excavations that were taken in the 1970s in the context of hearths that were identified throughout the stratigraphy. And we were able to recover ancient hominin mitochondrial DNA from 3 of the samples that were taken. And I'm here just showing you sort of these plots, showing you the elevations of C2Ts to show you that sort of we were able to authenticate that the DNA was really ancient. And although for 2 of the samples coming from layers L11 and L12, we didn't have enough data to tell which type of ancient hominin this was, we did have enough data from layer L14, so that's an early organization layer, to be able to attribute the DNA that we recovered to ancient human mitochondrial DNA, which, as I'm sure many of you know, is what you would mostly expect in an organization context. And so this showed that it is possible to apply this methodology also to look for ancient human mitochondrial DNA, which can be trickier to recognize because it's more similar to the contamination that we are introducing during excavation and lab work. Now, one of the possibly, at least for me, more exciting aspects of research that we could think of to apply this methodology to look for ancient hominin DNA at archaeological sites was to look for Denisovan DNA outside of Denisova cave. So I guess many of you heard that Denisovans, these distant relatives to Neandertals, were first discovered in 2010 based on the genetic evidence of a sample of a toe bone that was found, finger bone that was found at Denisova cave that I already mentioned. And until very recently, the only evidence we had of Denisovans was at that site. So we have this bone and a couple of teeth that have been identified as Denisovans, as well as the Denisovan DNA that I mentioned in the sediments. But we also know from looking at modern genomes of people living today that some people carry Denisovan DNA in their own genome, particularly in Oceania. And from that, it was inferred that the range of Denisovans in the past must have been quite large, possibly where I'm showing you this red, this vague red range. It's possible that Denisovans were inhabiting large part of Eastern Eurasia. But we had no evidence of them outside of Denisova cave. Now in 2019, there was a study coming out of a site in the Tibetan plateau at Baishia cave, where a half of the mandible, this is a picture that is reconstructed, half of the mandible was identified. And unfortunately, there was no ancient DNA in that sample, but the team was able to recover protein sequences from that mandible. And based on the proteins, they were able to infer that this individual came from either a Denisovan population or something related to Denisovan populations. But again, this was something that we did not have direct comparison to the Denisovans in Denisova cave because we did not have any DNA from that mandible. And in another study that looked at the same site, and this time looked into sediments that was sampled throughout the stratigraphy of the site, 6 of the samples that were collected, sediment samples from 6 of the layers that were sampled were shown to be positive for the presence of ancient mammalian mitochondrial DNA, following this same strategy that I talked about of looking for general mammalian mitochondrial DNA in them. And these are sort of the layers that are marked here in green. And out of these, 4 of the layers marked in the red stars also contain ancient hominin mitochondrial DNA. And quite excitingly, there was enough DNA to reconstruct, at least for one of these layers, a full mitochondrial DNA genome. This is from layer 4. And it was shown through different analyses that I won't go into the details of, that the sedimentary DNA that was recovered all fell within the variations of Denisovans, and specifically within sort of the most closely to what was known as the later Denisovans from Denisova cave, showing, based on now genetic evidence, the presence of Denisovan-like populations at this Tibetan site at least 100,000, 60,000, and 45,000 years ago. And it is my hope that we will be able to apply this at other sites and sort of find other traces in other places in time where Denisovans were to better understand their trajectory throughout the world. One of the other things that has been happening lately is taking this methodology and simply scaling it up. So one of the things that has been happening in the field of ancient DNA is the automation of a lot of the lab work. So going from preparing samples manually and sort of pipetting and doing all the steps to putting this on pipetting robots in 96 or even 384 plates. That means you can process in parallel and in a very reproducible way much bigger amounts of samples. And that allows to do analyses that are a much bigger scale than was possible before. So to show this, I'm coming back to Denisova Cave. This is a study that was led by Elena Zavala and Matthias Meyer in Leipzig, where they sampled with their collaborators 728 sediment samples from just from Denisova Cave, spanning all the layers from the Middle Paleolithic up until the Upper Paleolithic. And then they applied similar methodologies to the ones I've shown to look for ancient mammalian DNA first, and then specifically ancient human DNA. And this is a result of the faunal DNA, of the mammalian DNA. And you see, I'll walk you through it. It's a bit of a complex picture, but you see here stratigraphies of the east and the main chamber. The east is the one that I've showed you before from the pilot study. And each of these sort of pie charts is a sediment sample that was taken. And you see in each of the pie charts the different taxonomic identifications that were made. And here is sort of the types of taxa that were found. And the first thing that is sort of a take-home message from this figure is that almost all of the samples contain ancient DNA. The success rate at the site and the preservation of DNA at the site is quite astounding, with nearly 95% of these samples containing ancient DNA. And this really allows to do very in-depth analyses on population genetics of fauna and also potentially humans at the site. Now, the second thing that we can start to look at is the differentiation in the taxonomic diversity over time, and that is over layers. So in the lower part of the stratigraphy that you can see both here in this chamber and here in this chamber, you see a larger predominance of green color, which is ursids. So there's more bare DNA when you're in the older layers of Denisova Cave. And as you start moving up the stratigraphy through different layers, you start seeing sort of the bares, the green sort of fading out, and more of the sort of beige and brown, so the bovids and the hyenas are getting a larger proportion of the DNA that is found. And that is actually fitting very well with what is known from the zooarchaeology analyses of these sites, where there is also a larger proportions of bears that are found in the Middle Paleolithic layers further down, and then there's a switch as you go up to far more evidence for hyenas and bovids and cervids. And that has also, of course, has to do with climatic changes. And that was also sort of a good representation that the DNA that is found in the sediments corresponds well to the zooarchaeological record. Now, what about humans? So these are the same two chambers, the east chambers and the main chamber. And now you see the results of the hominin mitochondrial DNA. And of course, the success rate of looking for hominins is lower, but we're still at 25% success rate. So 25% of the samples that were taken yielded at least some ancient hominin mitochondrial DNA. That's more than 100 samples, and that allows to do a lot of analyses on the specifics of these ancient hominin populations at that site. So again, to walk you through some of the main outcomes of this study. So the green sort of background of the layers, that's the Middle Paleolithic layers, and the sort of more orange pinkish at the top, these are the initial Upper Paleolithic and the Upper Paleolithic layers. And in these, in the spots, so in the sediment sample, if the sediment sample is marked as white, it means there is no ancient hominin DNA. And then if it's red, it looks Denisovan. If it's blue, it looks Neanderthal. And if it's yellow-orange, it looks modern human. So one of the first things that one can see is that the earliest hominins to appear at Denisova caves, so the lower in the stratigraphy, are the Denisovans, with Denisovan DNA found in association with the early Middle Paleolithic tradition, and arriving at the cave around 240, 250,000 years ago already. And now more specifically within the Denisovan mitochondrial DNA, we can see that at the bottom, there is this one type of Denisovan mitochondrial DNA that was marked here as a arrow pointing down. And this is the type of Denisovan mitochondrial DNA that fits also the older fossils that were found. And as we go up the stratigraphy and sort of later in time, here you see the ones that are sort of these arrows of the older type of mitochondrial DNA of Denisovans. And as you move to layers that are older than 80,000 years, you start seeing a change in the types of Denisovan mitochondrial DNA here, here, and here, where we now have a switch, and what we see is the later type of Denisovan mitochondrial DNA that fits also what is known and what was seen in the site in Tibet that I showed you, indicating that after 80,000 years, there was a turnover in Denisovan populations, and that this type of later Denisovans became what is more, apparently, what is more predominant, not only in Denisova cave, but also all the way into Asia. So that's the Denisovans. Then we start seeing that the Neanderthals only appear later in the cave. As I've mentioned, the one sample that was coming from layer 14 from the pilot study turned out to be redeposited from somewhere higher. And so the first Neanderthals start to appear later, only about 170,000 years ago or so. And then we have several episodes of sort of Neanderthals and Denisovans interchanging, or sometimes being in the same layer and even in the same sample. Also with the Neanderthals, there was an interesting case, as one of the samples, one of the earlier Neanderthal samples that were discovered, turned out to be from a lineage that we had not sequenced before. So we can tell, or they could tell, that it's closer to Neanderthals than it is to Denisovans or to humans, but it did seem to be a distinct lineage of Neanderthals that had not been known before from skeletal remains. And then lastly, we come to the humans, where we start seeing human DNA, mitochondrial DNA, in the initial upper and the upper Paleolithic layers. In the east chamber, in this area, there's a lot of mixing, and indeed we see a bit of a mix of both of all three types of humans. In the main chamber, where the stratigraphy looks to be cleaner, there's a predominantly human mitochondrial DNA, showing that modern humans arrived to that area at least 45,000 years ago. Okay, and then for the last part of the talk, I want to mention that everything that I spoke about so far in the archaeological context has been mitochondrial DNA. Now, mitochondrial DNA is very useful when we look into ancient DNA studies, because it's present, it's a smaller part of our genome, and it's present in many copies per cell. So if ancient DNA is preserved in an ancient sample, it is more likely that we'll be able to find mitochondrial DNA than as we first screen for ancient DNA. But mitochondrial DNA is only inherited from a single lineage, from a mother to all her offsprings. So if we're working on mitochondrial DNA, in reality, we only ever get sort of part of the picture of the population history of an individual, and the individual's ancestors. To get the full population history, what we need to do is look at nuclear DNA. That's the DNA that is present in all of our cells in two copies. One we get the copy from mom, and one we get a copy from dad. So it was becoming quite clear at this point, which was actually not very long ago, that sort of the next big step would be to move on from mitochondrial DNA into the realm of human nuclear DNA from sediments. And I want to show sort of first this study that came out quite recently, where there was a single sediment sample that turned out to be quite spectacular from a site in Western Georgia. And this is a sediment sample dated to 25,000 years ago that from just by shotgun sequencing, that is without any of the capture that I talked about, just extracting the DNA from the sediment and sequencing as it is, the researchers from this project were able to recover genome-wide nuclear data from a human, a wolf, and sorry, my slide popped, a wolf and a bison. And they were then able, by looking at this nuclear DNA that they recovered, to look at the population history of each of these three taxa from a single sediment sample. So for example, without going into the details of these colorful plots that indicate ancestry, in the sediment sample, the human nuclear DNA was found to be from matching more closely to a pre-late glacial maximum lineage that had substantial ancestry from what is called these basal Eurasian population that had a lot of impact into the human genetic makeup of the humans in the past and also today. The wolf turned out to be from a previously unknown extinct lineage. So again, another example of taking DNA from sediments and discovering lineages that hadn't been known before from sequencing skeletal remains. And the bison turned out to be basal, that is ancestral to all the populations living today. So one sediment sample inferences on three different taxa. Now, another way to do that is to again, look at this possibility of doing capture. And this is from a work of Ben Verdon and again, Matthias Meyer, where they designed a capture probe that would look at 1.6 million positions in the genome, in the nuclear genome, that would be specific to humans, but also would be informative of the ancestry of humans. That is, allow us to differentiate between ancient human groups and ancient hominid groups. So just to show you how this works, they first took a dummy example, let's say, from skeletal remains, and then I applied their methodology to see if you do this on skeletal remains where you know what to expect, if this is Neandertals or Denisovans, you're still able to recover it from this capture methodology. And what you're seeing here is on this axis, the x-axis, as you go more towards the right, you're more likely to be a Neanderthal, and as you go on the y-axis higher up, you're more likely to be a Denisovan. And then when this was ran on skeletal remains of so low coverage genomes of Neanderthal skeletons, it was found that they all fell sort of within what you would expect. So having a higher proportion of Neanderthal DNA. And when you ran this on Denisovan skeletal remains, genetic data from Denisovan skeletal remains, you found a higher proportion of Denisovan DNA. And then this individual that falls in the middle is Denisova 11, and she her genome was sequenced, and she was found to be the daughter of a Neanderthal and a Denisovan. And so that's why she falls pretty much in the middle. So this was a good sanity check that the methodology could work to tell apart Neanderthal DNA from Denisovan DNA using these positions in the genome. And so then this was applied to quite a large array of sediment samples taken from 3 sites: Denisova Cave, again, Chekilskaya Cave, also in Siberia, and Estatuas Cave in northern Spain. And just to walk you through, beneath the sediments from Denisova Cave, all of the sediment samples that had Neanderthal mitochondrial DNA also showed nuclear DNA matching Neanderthals, whereas the sample that contained Denisovan mitochondrial DNA looked to be more Denisovan. And in Chekilskaya and in Estatuas, all of the sample looked to be Neanderthal, also on the nuclear level. Now, more perhaps more informatively or more interestingly, by looking at a large number of sediment samples, as you can see here in the dots, over the stratigraphy of this statuas cave, spanning quite a large time range in northern Spain. From looking at sediment DNA, both mitochondrial and most importantly nuclear, they were able to tell that there was a turnover in Neanderthal population, so a replacement of Neanderthal population from an earlier population that lived up until about, where's my mouse? Up until about 100,000 years at the cave, and then everything above that starts looking at like another population, a later population of Neanderthals. And this matches also an idea that they were sort of two splits or two radiations of Neanderthal population, an older type that is shown here in red that sort of splits from the common ancestors of Neanderthals about 135,000 years ago, and then a second radiation, a second expansion, about 100 to 115,000 years ago, who may have given rise to what is then known as the sort of classical Neanderthals in Europe. Okay, so that was the case study showing that it's also possible to look into Neanderthal and Denisovan population history from nuclear DNA from archaeological sediments. So overall, I hope that I was able to show you and convince you that by using this methodology, it is possible to retrieve ancient human, ancient hominin DNA from sediments deposited in archaeological context. So far, the oldest that we know of is some samples that are about 240,000 years old from Siberia. But we also know now that it's possible to do this from old collections. I showed you the example of the samples that were collected in the 70s, and also for more difficult contexts in terms of harsher climates for DNA preservation. We're able to identify a variety of taxa, including taxa that are now extinct, both on the faunal side, but more perhaps interestingly for our purposes, also including all three known human groups that are whose genomes have been sequenced, so Neanderthal, Denisovans, and ancient modern humans. We can now do, at the level of population genetics analyses on these ancient hominins, even when they have no physical remains at the sites. And so overall, I would argue that this is a highly parallelizable and also a non-destructive way to study ancient genomes over a large array of times and places. And with that, I'd like to thank all the people that were involved in the work that I showed you today. And I'm only mentioning the people that I have personally worked. Not everything that I showed today is my work, not at all. I'd like to thank the organizers in Liverpool for the opportunity to speak to you today. Thank you for your attention. And I'll just mention that since I'm a new PI, I'm also hiring. So if there's students out there who are interested in looking into sediment DNA, please get in touch. Thank you very much.
Cool, thank you, Viviane. So there's a couple of questions in the chat already, so we shall move swiftly on if that's okay with you. So Walter has asked, what are the geologic characteristics of a site to sample for DNA? And then following on from that, how long and how involved is the biochemical analysis cost? And can you speculate whether the hominins had a dietary preference for one animal or plant?
Okay, so I'm going to go one by one because then otherwise I tend to forget what was the question. So about the geological aspect, it's a really good question that I wish I had sort of a very decisive answer on. One of the things that I've been doing in my postdoc, but we don't still have the final analyses for that, is collecting sediment samples from sort of various contexts and trying to compare and contrast DNA preservation at different times and different places in different contexts. We know in general that sort of extreme pH, for example, is not good for DNA preservation. We know that from bones, and we've also seen that it's not great for sediments. Overall, as to context, this seems to work better in cave sites. If you compare that to rock shelters or open air sites, under the assumption is that cave sites have sort of a more protected microenvironments and are less disturbed by sort of temperature variations and other factors that could affect the DNA preservation over time. But it does not say, that's not to say that it doesn't work in other contexts. So we also have successes in rock shelters and open air sites. It's just cave sites, particularly in sort of temperate climate, the late Pleistocene, we have very, very high success rates. The second question, I don't know if I'm jumping to the third, was about the dietary preference. Yeah, I mean, I think that's definitely a possibility that a lot of the fauna that we see in the genetic signal comes from sort of what animals the humans brought into the sites. And I think that's true in many cases. Also, when people look into the zoological assemblages, particularly the ones that have like signs of having been butchered. So if I just take Denisova cave as an example, sort of woolly mammoths could not have possibly walked into the cave. It just doesn't, they just wouldn't fit, right? So they were either brought in by humans or they were brought in by other predators, so for example, the hyenas. But it's an interesting question. To which extent can we sort of correlate the faunal DNA that we see with the human behavior? We haven't done much of that, I have to say, but I think it's a very interesting thing to be looking at in the future, sort of how can we use these data to better understand the impact that humans had on their environment and how they use natural resources around them over different times. And I think I'm missing a question. Maybe Karis, you can remind me.
Yeah, so there was one in the middle, and it was how long and how involved is the biochemical analysis, and then comma cost question mark.
Cost is always a good question. Okay, so let's put it like this: the sampling at the sites is not very complicated in the sense that it's great if the person that is sampling at the site, whether it's sort of a geneticist or it's often done by the archaeologists themselves, since they know best their sites. It's great if people can sort of, put on a face mask and put on gloves and sort of try to use sterile tubes to collect the samples, but it's more being cautious about sort of not, coughing into the tube more than anything else. So that's not very involved, I would say. Then the DNA extraction, library preparation, capture, sequencing does take a while. So if you would do this sort of manually on a small set of samples, it could easily take like a month of work. Then can take very much longer when you sort of have to do repeated experiments. So I would say usually the timeline is that from the time you get the samples until you have the first results is about 3-4 months, something like that. The analysis, also takes a little bit of time. You have to sort of do the raw processing of the data and then look into which taxa you have in each library, then authenticate. And then, of course, once you have something interesting, then you want to look deeper into it, do phylogenies and things like that. In terms of cost, that varies a lot. I don't have a good number to give, but I don't know, a few $100s, but I don't have a good number to give.
Okay, cool. So there's another question in the chat from Nida, who says, is there any evidence from the DNA to show whether the earlier or the later Denisovans were more closely related to Neanderthals, i.e. or introgression events, or not?
That's a good question, and it's not something we can look at through mitochondrial DNA. So to look at introgression and sort of mixing between populations, you really have to look at nuclear DNA. And the nuclear DNA work at Denisova Cave was mostly done as a test case, and it was not sort of the main part of the study. But I'm pretty sure there's a follow-up on that. So I don't think I don't have the data to answer that question right now.
Does anyone else have a question? By all means, feel free to ask out loud.
I do, actually.
Yeah, go ahead.
I have a couple of questions. So first, thank you for your lecture. Like, it was very interesting. You're kind of my role model in life. So anyway, Could you explain more about the capture? Like, do you use primers to do so, or I
Yeah,
mean,
so
what the method is for capture?
okay, so this is a bit technical, but it's an on beads capture. So what you do to prepare the capture probes is that you take the mitochondrial genomes that you're interested in. In this case, mitochondrial, doesn't matter, it can be whatever else. You sort of take all the genomes you're interested in, and then you chop them into pieces. The probes that I've been using are 52 base pairs long. So you can sort of take this 52 base pairs, and then how do I say this? You sort of attach them to a magnet. And then your probe that's on a magnet, you sort of add them to the tube where you have all your DNA. And then because of similarities between the two strands of the DNA, if you have any DNA fragment in your tube that sort of looks or is similar enough to your probe that's on the magnet, then it will get attached to it. It doesn't have to be a perfect match. There's some sort of wiggle room, some errors, so it doesn't have to be 100% exactly the same mitochondrial genome, but it has to be sort of similar enough. And then once you have all your fragments that are attached to your magnetic probes, you can sort of use the magnets to sort of fish out physically and remove those fragments from your library and then put them in another tube and only look at those. I hope that answers your question.
Yeah, it does. Thank you. And I had another question, and I can't remember it right now.
We can come back to you if you like in a bit.
Oh, it's fine. I do know. Do we know, like, what did, I mean, where exactly the DNA comes from? Like, maybe from tiny bits of bones or coprolites or stuff, or?
I love that question because the answer is we don't know.
Okay.
So there's, a lot of hypotheses out there. Definitely what you said, sort of tiny pieces of bones and teeth that sort of are so ground up, they just become part of the sediment. It's definitely an option. Coprolites that are sort of also disintegrated and become part of the sediments. There's been in the literature on the sort of natural environment DNA, people talk about hair or feathers, depending on what you're looking at, sort of skin cells that are being shed. Urine is certainly an option. We don't know yet for each sediment sample sort of what it comes from. And I think this is a really important point because when we want to then later on go into deeper and sort of interpret human behavior, for example, if you're thinking like spatial organization in a cave or something like that, then it will become really crucial to know where the DNA comes from. But we don't know yet. We do know that within a single sediment sample, in some cases you have DNA from multiple individuals, and in some cases you have DNA from only one. And so if it's only one, then perhaps it's more likely it's a piece of bone or a piece of tooth. And if it's multiple, then maybe it's another type of, I don't know, tissue decomposition of body fluid or something that all got sort of mixed up. So it's all possible.
Okay, thank you.
Catherine, you've got your hand up.
Yeah, thanks. Hi. Thanks, Viviane. That was amazing. I learned such a lot. I wanted to kind of ask you what the scope was for can you go back more in time, kind of pre-Neanderthal? What's the scope for the lower Paleolithic or lower Paleolithic sediments? Or have you just not kind of done that yet?
I think low Paleolithic is going to be tricky. So I can only say, comparing to what we know from DNA in bones, sort of in permafrost so far, the older DNA that's been sequenced is actually quite recent. It's from mammoth remains that are over 1 million years old. But that's sort of an extreme. And when you go out of permafrost and are in temperate climate, then the oldest DNA we know is from Cima de los Suesos, so that's 430,000. And that's sort of deep within the Cima cave. So I don't know about, possibilities older than that. Possibly if you're very lucky and you find a very good preservation spot. But I think this will be more widely applicable to sort of middle Pleistocene, late Pleistocene, Holocene context, in a similar way to DNA preservation in bones, essentially.
Okay, great. Thank you.
Okay, so I guess last question is from Hannes in the chat, who says, I wonder if older contamination can show ancient DNA pattern. Can we differentiate ancient DNA from a trace DNA from an archaeologist who touched the material, for example, 30 years ago?
Yeah, that's a great question. So 30 years ago is not so concerning. What has been shown sort of experimentally by looking at damage patterns, again, this is bones, not sediments, but we think in many ways these are comparable, is that when you take DNA from sort of modern people, you get these C2T damages that I talked about, up to 5% or something like that. And then as soon as you go back, 5,000 years, you start seeing this in much higher extent, so 10%, 20%, 30%. And yeah, the DNA damage accumulates, but it accumulates over time. And we don't think that 30 years is enough to sort of tip the balance towards us thinking that it's ancient. So yeah, I don't think that sort of even excavations 100 years ago, I wouldn't be too worried about this sort of showing up as false ancient.
Okay, cool. So we can let you go now, Viviane. But yeah, thank you for a really good talk. Thanks everyone for coming. I hope that we'll see you all again next week. We're having a talk by Xixia Yang from the Chinese Academy of Sciences. Her talk is going to be called Case Studies on Environmental Change and Behavioral Adaptations in North and Northeast China. So that should be really interesting. So yeah, once again, thanks everyone for coming, and thanks Viviane.
Thank you so much.
So yeah, bye everyone.
Bye, thank you everyone for being here.
Thanks, Viviane, that was amazing.
Thank you.